Good morning. Welcome everyone to the IIVS-sponsored webinar, How GLPs Enhance the Quality of Regulated and Non-Regulated In Vitro Toxicology. Today's webinar will introduce concepts of GLPs designed to promote study and data integrity within an in vitro toxicology framework. A 15-minute question and answer session will follow the presentation. Before we get started, let's take a moment to ensure that everyone is familiar with the webinar toolbar and can access important aspects of GoToWebinar. Due to the large number of attendees, all audio has been muted except for our speaker and will remain so for the duration of the webinar. You may ask questions by using the question feature at the bottom of the control panel at the right. Click on the arrow to maximize the control panel and view the question window. Questions can be submitted at any time, but will be held until the end of the presentation. We are recording this webinar and the question and answer portion. A PDF of the presented slides and a link to the recording will be distributed to the webinar participants in about 2 weeks. Now it is my pleasure to introduce you to our speaker, Amanda Ulrey, who is the Director of Quality and Compliance at IIVS. I will now pass the controls over to Mandy, who will begin the presentation.
Okay, thank you, Laura. Good morning and good afternoon to everyone who's attending. As Laura mentioned, my topic today is how GLPs enhance the quality of both regulated and non-regulated in vitro toxicology. And really, in general, it's all types of toxicology, but here at IIVS, we focus on non-animal test methods. So many of my examples are going to be pulled from in vitro toxicology, although it can be applied to any type of laboratory work using animals or otherwise. So good laboratory practices, or GLPs, are required to be followed for any work that's being submitted to the regulatory agencies. So if anything, any of the work that you're doing is part of any data submission package, GLPs are a non-negotiable. It has to be followed for that. But what I'm hoping to do in this presentation is to make a case for using certain elements of the GLPs to improve laboratory work in general, whether your end goal is submission to the agency or not. I believe there are elements of the GLPs that can bolster quality of scientific research throughout. So recently, there have been a number of editorial articles in both Science and Nature regarding the lack of reproducibility currently in published scientific work. As you can see from this graphic that's from one of the Nature articles, 90% of the researchers that were surveyed felt that there is some problem, either significant or a slight problem, currently with the reproducibility of published work. This is concerning. When scientific work is not reproducible, a large amount of time and resources are wasted. Reputations of scientists are damaged when published work later turns out to have been false or have problems and has to be retracted. And then if there's any media coverage for the work, any kind of public opinions or even scientific opinions then have to be swayed and turned later when you have to retract the work once it's published. So in the Nature article, researchers were asked what factors they felt could be put in place to boost the reproducibility of work coming out of the labs. There are a number of things that they mentioned that GLPs can really in no way touch on, like journals enforcing quality standards and receiving incentives for better work or receiving incentives for doing formal reproduction of someone else's work. So GLPs don't touch that, but the topics that I have circled here, where there is mentoring and supervision robust study design, better teaching, more time for men, more time checking notebooks. All of those items that the researchers felt could contribute to boosting reproducibility are actually talked about in good laboratory practices. So as a quality professional, that made complete sense to me. What was surprising to me is that at the same time, there was a paper that came out in the Society of Toxicology in their publication, Toxicological Sciences, titled, "Does GLP Enhance the Quality of Toxicological Evidence for Regulatory Decisions?" And this paper specifically was focusing on some endocrine disruption work, and there are a number of researchers who seemed frustrated over GLP compliant work and data being weighed more favorably than studies that were conducted under non-GLP conditions but were run by experts in the field. So this paper sort of dissected the differences between what the experts in the field brought to the study, as well as what GLP compliant work brings to similar types of studies. And the paper noted that in order for a study to be GLP compliant, certain quality standards must be met, and that these standards generally boost the understanding and transparency of the work that took place in those studies. What GLPs do not do is take the place of a knowledgeable researcher. So it still is important to have really scientifically strong individuals in charge of work, but there is something to be gained by running work under GLP standards, and that should help in making any kind of work coming out of a GLP environment more regulatory sound for people to look at and review. So, do GLPs enhance the quality of toxicological evidence for regulatory decisions? Well, yes, I think they do. So here is a hypothetical in vitro toxicology lab. They're doing some non-animal-based work in this hypothetical lab. This came out of a different Nature article titled "How Quality Control Could Save Your Science," and I do have a link to that article here in the presentation. And we'll go through and use this lab as our example for how following a GLP quality system can improve the work that these scientists are doing. In vitro assay development, if you're looking at method development, generally you start with a large number of prospective new methods and ideas and work with them and pull your best forward into a research and development R&D environment. And then some of the less robust and less sound methods will fall to the wayside, but hopefully you'll discover a number of methods that look really promising that you might want to do some additional work on and refine farther and do some publications and presentations, oral presentations, poster presentations based on that work. And then from there, there will likely be additional studies that, again, fall by the wayside as the more robust are picked up and moved forward into more formal validation studies, where you do some work on reproducibility, inter and intra-lab reproducibility, and look for false positives and false negatives, and really test the assay to see how it stands up. And then eventually, if you have a really solid assay, those assays move forward into regulated use with OECD test guidelines created for them and wide industry acceptance. This funnel also is applicable to how novel chemistries and new materials move through their development pipeline. You'll start with a number of ideas and slowly take that down until you get to your best materials to move forward. What I would like to propose is taking elements of good laboratory practices and breaking them up and starting to apply them earlier in the research and development process, and some additional ones at the point where you're trying to publish a method, and then having additional controls to put in place when a method's moving forward to validation, instead of waiting until you have an assay that's accepted for regulated use and then retrofitting a quality system. I think there are a number of improvements to the research and the work that's coming out of a lab that you'll appreciate, and you'll gain additional confidence in what's coming out of your lab as you apply some elements of good laboratory practices earlier and earlier in that pipeline. Even as early as the basic research and development phase. GLPs for regulated studies are not required for any work done in an R&D environment, but I believe there are concepts that can be applied even in R&D that as researchers you will appreciate benefits from and improved quality. The first being good documentation practices. So I have excerpts here from the FDA GLPs that talk about the requirements for GLP compliant data collection. On a very simplified, basic level, what you're shooting for in GLP compliant documentation is making sure that the study data are accurate, that they are legible, that they are contemporaneous, original, and attributable. And that is what that acronym ALCOA stands for. Those are all the elements of good data collection that should be in place, even, I feel, at the research and development phase. So ALCOA calls for documentation to be original and recorded directly and contemporaneously. So what this means is you don't want to write any data down on non-lasting media, like a Post-it note or the back of your gloved hand. Take a look at your R&D environments and see if there's any way that you can modify your data collection to make it easy to collect the data directly to the place where you are going to be saving it. So directly into your lab notebooks or directly into forms or worksheet pages. So whatever system you have set up for data collection, you want to make sure that it supports original data being recorded directly and contemporaneously. The other thing that needs to happen with all data is it needs to be attributable. You need to be able to tell who has made the entries. One reason is so that if any additional details are needed, you can find the person who made the entry and figure out what exactly happened and what additional details need to be added to the documentation so that it can stand alone. But another reason is that if the data are attributable, then the person that's making the entries really takes a sense of ownership of the work that they're doing. So all entries should be initialed and dated, and it should be assured that whatever is being signed for is clear, that you can clearly tell what an individual has done or what the initials on the bottom of the page are there to document, which action they're tied to. So going back into our hypothetical lab, situations like this where there is a notebook and data is being collected in the notebook, but it looks like there's also some important information that's floating next to the notebook on Post-it notes and written on a paper towel. That should really be avoided. You want to capture everything directly into your notebook so that you do not lose any critical information. And I feel this is important whether you're in an R&D environment or in a GLP environment. Another quality concept that could be important for research and development is to have standard laboratory procedures. So there is a way to tell and knock out some of the causes for potential variability. So if you have a standard way that incubators are maintained and you know what temperature the incubators in the lab are always set at, then you'll know that if something happens with your cells that are in the incubator, hopefully it's not because the incubator temperature was higher or lower than what you thought it was. If everyone working within the lab, several different researchers, several different lab staff doing different projects, if there is a standard way that all your equipment is maintained, then everyone should be aware of what those standards are and can work to those standards and know that for their work, that's what the temperatures were set at, that's where the humidity was, the CO2 was controlled in a certain way, and just to really control as many variables as you can in the work that you do. I think standard laboratory procedures are helpful. Another benefit if you have standardized laboratory procedures is that if you know for the work that you're doing that you need to do something that's against those standards, then you can reserve the equipment for your specific use. You can make sure nobody else is fiddling with your equipment settings or setting it to what it normally should be if that's not where you need it for your work. But before you can do that, you need to know how things are normally handled. Also, you would know there are a number of tools as scientific methods become increasingly more sophisticated that can be used in a variety of different ways. HPLCs, flow cytometers, even plate readers for optical density. There are different ways that individuals could be used to getting data from those data collection machines. And if you're a researcher and you need things to be done in a different way, then you may need to provide additional instructions for the laboratory staff that is actually using those machines for you. So you would need to have first standardized procedures that you know that they're following so that in the future you would know that you need to provide additional guidance so that they do what you are hoping to do. So again, back to our lab, in this picture, we have an individual who's using a piece of equipment even in a research and development environment. You would want to have some sort of understanding of how she's using that equipment. Another quality control element from the GLPs that can be very helpful in a research and development setting is to have some control over your media and reagents. The GLPs state that all reagents and solutions should be labeled to indicate the identity, titer, concentration, storage requirements, and expiration date, and that you should not use any outdated reagents. There are a number of materials that you could use in the lab that they have expiration dates because an active ingredient is no longer stable beyond that date. So you wouldn't want to think that your media requires certain supplementation based on what you thought you were using, when in reality the active ingredients for whatever media supplementation you were using could have expired months ago, and you don't need that specialized media or reagents. Heading back into our lab, there are two instances here. One is the best before 1985 expiration date. Obviously, you want to make sure that you're using current media and reagents and you don't inadvertently use expired reagents. Two other areas on in this lab picture are up here towards the top right, where we have a number of test tubes. Something's happening in those test tubes, maybe a serial dilution, but those test tubes don't appear to be labeled. So if those researchers need to hand off the work to someone else, there's no guarantee that the other person coming in to take over would know what's going on. Also, if you're doing work one way on the rack of tubes and you pick it up in a different orientation, if your tubes aren't labeled, you could end up with your whole serial dilution being dosed backwards. So you definitely want to label everything so that you're sure what it is that you're working with. So those were the methods and parts of GLPs that I think can be implemented in a research and development, strictly research and development framework to help aid in reproducibility of the work in the lab. There are additional controls that I think can be put in place once you have been working with a method for a while and you're getting ready to go public with it and publicize it in a peer-reviewed journal or in podium presentations or posters. The first GLP quality concepts, the first two actually, I think go hand in hand for research intended for publication, are the use of a protocol and the concept of having a study director. So using protocols, protocols are predetermined study plans that are approved by the study director and are written that clearly indicate all of the methods for the conduct of the study. When you're determining your protocol ahead of time, you're figuring out what equipment needs to be used, what media needs to be used, all of the critical time points for dosing, for culturing cells, everything that's critical is written down in your methods for your protocol. The other powerful piece of a protocol is including information on how you're going to do your data analysis. So setting that out ahead of time really helps to control against unintentional bias in your research. And then as a researcher, you can provide the individuals working in your laboratory with the protocol so that the laboratory staff have precise written instructions for what they're going to do, so that as a researcher, you don't have to stand behind them and watch everything that they're doing in order to have confidence in the work that they're doing. They have instructions, and then they will follow good documentation practices. And those two pieces together should really help for pulling together later manuscripts or posters. So you really pretty much have everything that you need pre-written with the exception of your results. And the other concept that I had mentioned that I think is very powerful and goes hand in hand with the protocol is the concept of the study director that comes right out of GLPs. The study director is the single point of control for the study. So they have the authority to go against the plan if they need to. This puts the power and the knowledge with them. So this is your most knowledgeable researcher. And although you have a protocol, this is science, so things don't always go according to plan. Having a study director gives you your go-to person for when things do not go as expected. The study director can verify that although things have not gone according to plan, that it is okay, or they make the determination that it's not okay, and you go back and redo work. They also have the ability to modify the plan when something unexpected happens. If you're testing some materials that need to be, they're very stubborn, say, and you're dosing a three-dimensional tissue construct, and they just will not rinse off under normal protocol conditions, the study director is the person who has the authority to say, okay, maybe you need to wet a Q-tip and a swab and go in and see if you can remove it that way. This gives the power concentrated to one particular person to be able to do that, which, again, is very important if you're moving towards publication for your work. This is the person who's controlling all of those variables so that you don't end up having to repeat additional work unnecessarily just because procedures weren't followed the way that you would have liked them to be. So back in our hypothetical lab, we have two individuals. We already noted that they are not using labeled tubes, but I don't believe that they're following the same protocol because one of them is doing his dilution on ice and the other is at room temperature. If that is a variable that affects the study, then that information should be controlled in some way. It should be written down as an instruction in the protocol, whether this should be done at room temperature or on ice. So we touched on this a little bit in the last slide when we were talking about protocols, but in addition to having all of your statistical methods and acceptance criteria, in addition to having your statistical methods written down, in your protocol, you can also have predetermined assay acceptance criteria. This would be information that you are putting into the protocol that will give you, the researcher, confidence in the data that you are receiving at the end of the study, so that you try to control for as much variability as you can in test systems over time and within the same lab. With in vitro assays, you could have a number of different types of test systems, from ex vivo tissues, like here, number 1, that's a bovine corneal opacity and permeability assay cornea holder. So there's an excised bovine cornea on the inside of that. Number 2 is an example of a three-dimensional tissue construct. Number 3 is a test system that is actually just a chemical matrix. And 4 and 5 are monolayer cell culture test systems. In the in vitro world, you could run into all of these different types of test systems. So in your assay acceptance criteria, you can set out the parameters for how you know that this biological test system is acceptable for use on this day that you're doing your research. We have the luxury, since we're not having to justify the use of animals in an in vitro setting, we can use concurrent positive and negative controls to help us have a level of confidence in the biological test systems that we have that day. So we know that they're performing within an accepted standard range, and we can rely on the data that we get at the end of the day. So having those predetermined assay acceptance criteria as part of the protocol and as part of the information that's presented in your publications, I think that really strengthens the quality of your research and puts out some top and bottom baseline quality, so that if someone else is trying to reproduce your work, if they're getting different results for their positive and negative control, then they know that the data that they got that day does not correspond to yours. So those methods that are promising, show themselves to be promising in publication, then they might move into a validation trial, a multi-lab or single lab validation trial. And here I think there are a number of GLP elements, if not full GLPs, that could really benefit from being applied to all validation work that is performed. One quality concept for method validation is training. The GLPs require that every individual working on a study clearly understands their responsibilities and has the proper education and background to be able to perform their work. Even at this stage, you're working with protocols when you're doing method validation. Even with a well-defined protocol, there can be differences in technique that could affect the results. Here I have a graph that I've used before in our annual practical methods for in vitro toxicology training course to show the importance of training. So in the light blue, that is indications of positive control results for phase 1, and in the purple is phase 2. So 2 different runs of this protocol. It happened to be the same protocol run on 2 different types of cells. So on the left, we have 3T3 cells, which are fairly robust, treated in phase 1 in 3 different labs and phase 2 in 3 different labs. And you can see that those results are fairly standard. Everyone followed the protocol and got really nicely reproducible results. For the graph on the right, that is work with NHEK cells directly. And you can see that in phase 1, although all 3 labs were provided with the same protocol, 2 labs really held tightly together and had very similar results, but one of the labs was quite off, and they were following the protocol. But what we ended up doing in this instance was having a very intensive training where everyone got together and they really discussed the nuts and bolts of how to actually carry out the work in each protocol. And you can see once they did the training, again with the same protocol, labs 1, 2, and 3 in phase 2, the results were very similar. So they were able to really bring everything in line, not with just using the protocol, but by also having very specific training to the protocol that was provided. So a lot of times, method validation studies, multi-lab validation studies, are managed by a management team across a number of different labs. Different labs can have different standards for how they are controlling their equipment. So I think it's very important at the phase of method validation that, especially the management team, that they understand and be aware of the different ways that equipment is inspected and cleaned and maintained and used within each laboratory. There could be different methods of use or settings or calibration procedures that really are not acceptable for the way that the work that is being validated should be handled. So first you need to have those standardized methods in each laboratory, and then the management team needs to know what those methods are so that it can be determined if they need to be modified to participate in this validation work. So back in our lab, you can see in this instance, there presumably was a plan set out to do some kind of service on the equipment, but there is no actual record of that equipment. So when was the last time this piece of equipment was calibrated? That is something that could have huge impacts on the work that's coming out and the data that's collected from that piece of equipment. There are also pieces of method validation that could benefit from implementing some data integrity concepts. The MHRA recently came out with a GXP, meaning any kind of good laboratory, good clinical, good manufacturing processes. It all encompasses all of that, all-encompassing data integrity definitions and guidance for the industry. And it's a really well-written, in my opinion, guidance, although it's still in draft form at this point. But it introduces some things to think about concerning how your data is handled and where it's stored and what controls are in place to protect it all throughout the process pipeline, whether or not it's collected on paper or digital collection. If you have digital records, these data integrity concepts are the same. Some pieces from that guidance that I think a validation study would really benefit from is to control some of the conditions for data processing. For example, locking some cells on custom Excel spreadsheets and having specific instructions, including settings and parameters for other statistical processing programs that might be required to analyze data. The study management team is sending all of these data collection spreadsheets to different laboratories that are collecting data differently, plugging it in differently, and just putting in place some very simple controls at this stage to make sure that formulas aren't inadvertently changed and data aren't written over and that they're pulled from the original source. And thinking about some of those concerns at this stage, I think can help make the study management team's job of all that data compilation much easier, the more standardized that process is. Another data integrity concept would be if you are collecting data electronically, that those data are attributable to the person who has collected them. One way that it would not be attributable is if you have a common username and password logon for different programs. And I know that you get charged for how many licenses you have, by how many people are logged on, but from a data integrity standpoint, if you're using a common password and common username, it's much harder to control access to that program to only trained individuals, and you've lost your data integrity because you no longer have any record of who has actually collected the data. At IIVS, we've participated in a number of assay validations, and a few times some concepts have been applied at this stage for incorporating critical vendor audits into the work. Validation studies are large and costly, and they take a lot of time. And performing vendor audits of the vendors for maybe test system suppliers or data collection, if there's a new data collection machine or service, taking a look at those and performing a quality audit on those systems could be very important in controlling the work at this stage to make sure that moving forward, these test systems or this data collection system is going to continue to perform reliably because they being the manufacturers, have their own quality systems in place so that moving forward you can rely on them. This is a little spoke wheel of how you can approach a very simplistic critical vendor audit. What I think is a good place to start is right in the center, to look at either the lot or batch number of whatever test system you have or the version of the data collection system you have, and work outwards from there so that you can look at actual records at the vendor for training records for any of the personnel who have done any work on that, or equipment records for any of the equipment used in the production of that particular test system, or quality control that might be in place. This is especially important for data collection systems and anything with programming, to take a look at the versioning and quality control of applications. Documentation practices, obviously you're not really going to be able to get any of these other records if the vendor doesn't have good documentation practices. And then tracing everything back to source material records when you can, if it's a manufactured biological system. And then also archival procedures for all of those records and training records and quality control records, and versioning and code changes for electronic systems. All of that information can be looked at in a vendor audit and should really provide a level of comfort with the work that is coming out of those vendors and the products that are coming out of those vendors. So, and then the next step, hopefully, is that validations are successful and the new assays move into regulated use. And at that stage, the entire data integrity pathway should really be completed. So from the point of protocol generation all the way to data archival, looking at the integrity of the documentation, whether it's paper-based or electronic, and maintaining it in a reliable way all the way through, so that if you're tasked with showing those records at any point or defending your work, you have everything that you need at your fingertips. At this point, there should also be an independent quality assurance unit. Any regulated work has to fall under GLPs, and an independent quality assurance audit group is part of the requirements for any GLP work. And then at this stage, really, we've been working towards it all along, but the reporting structure within the facility should really be strengthened to make sure that there is facility management overseeing how the entire facility compliance is handled, and the study directors are in control of individual studies as they come through, and then the quality assurance unit serves as an independent audit function to look at the compliance of the facility and the individual studies. So in conclusion, I hope that I've made a strong case for how GLPs can be used not just as a regulatory requirement, but as a quality system, and that how they can be applied modularly to different phases of work to really help bolster the work coming out of a laboratory and the reproducibility of that. And I also wanted to mention too, if you are a new method developer and you're following this sort of modular quality rollout, hopefully the amount of work that you have to put in by the time you get to that regulated use of this work at the end is much less lengthy. So because you've been putting in place GLP concepts all along, so it should be very quick to switch over into fully regulated use. So going back in this awful hypothetical lab, when you're applying your GLP quality systems, whether you're doing regulated work or non-regulated work, all of these areas that have been circled would be taken care of and corrected and tightened so that all of the information coming out of the lab is of a high quality and much more likely to be reproducible.
Thank you, Mandy. Now we will open the floor up to some of the questions that we have received. Okay, the first one is, what type of training is needed for staff working in the lab? Okay, so training would be really dependent on the type of work you're doing. In a broad sense, if you're working in a fully GLP compliant lab, then you should have training on good laboratory practices for any staff working in the lab. If you're not shooting to be a completely GLP compliant lab, but you want to apply good documentation practices, say you're back in an R&D environment, I would recommend then at least training to those Alcoa data capture processes so that at least the researchers in the lab are pulling together their documentation in a way that's attributable and can be followed. So that's on a basic level. And then I would also imagine that individuals would be trained on the specific methods that they're performing. So I've seen modular approaches to training where individuals are trained on an entire protocol. I've also seen critical steps style training where individuals are trained on portions, small parts of different assays. So yeah, it depends, and every lab should train to their methods too. The next question we have are, how are GLPs implemented in industry? So that's a good question. We at IIVS, we are a CRO. So there are a number of different contract research organizations that perform work, just laboratory tests, and provide our sponsors with reports. And we function in a fully GLP compliant manner so that we are giving unbiased GLP compliant reports back to the sponsors, and then they submit them to the regulatory agencies. I do think some sponsors also perform their work internally under GLPs. Sometimes they're the ones who are making the submissions directly. Other times we're asked to follow GLPs even for studies that are not going to be submitted anywhere, just because it is a very good quality standard. So if you're working in the cosmetics industry or any other industry where you're not required to submit data to the agency, we at least have some sort of quality standard in GLPs behind the work that
those types of companies are doing. So they find that to be helpful as well.
Okay, another question. Do you have any additional comments on the draft revisions to the FDA GLPs?
Right. So one of my slides, I referenced the proposed revisions. For those of you who don't know, at the end of August, the U.S. FDA published a draft to revise the good laboratory practices. They have not been revised since the mid-1980s. So this is fairly significant. It's 141 pages, the draft revisions. So I'm still working my way through them, but in general, the whole concept of data integrity, I pulled that out of there. They reference concept of data integrity a number of times. They also are, there are a number of references to using GLPs as a quality system and using it to generate additional reports to management and that sort of thing. So the revised GLPs appear to be moving towards a GLP quality system. There are also a number of things that are discussed in the proposed revised GLPs concerning multi-lab studies and how to manage a multi-laboratory study. So those are definitely based on the GLP guidelines for multi-site studies, but that could have a lot of beneficial information if you're working on pulling together a validation study in particular.
Okay, the next question, is there any guidelines for GLP compliance?
There are lots of guidelines for GLP compliance. So there are global guidelines that can be followed that have been published by the OECD. There's actually one specifically that the OECD put out pertaining to the use of GLPs for in vitro assays. Also, each individual country has their own GLP guidelines to follow, at least one version. Here in the U.S., we have the Food and Drug Administration, the FDA, and the Environmental Protection Agency, EPA. They each have their own GLP guidelines. And then globally, I made reference to the MHRA. They have their own guidelines. So there are a number of different guidelines that can be followed. And maybe what I can do is when we send out the slides, I can add a page with links to all these different GLP guidelines because they can all be found online, and they're also valuable resources. I would encourage you, if you are looking at the GLP guidelines, to not just read the regulation, but at least here in the U.S., there's a lot of good information in the preamble to the GLPs. That's where, by our process, industry and different, academic institutions and anyone really has a chance to comment on any proposed regulation. And in those preambles, the FDA has answered some of those comments and explained additional rationale behind what the regulations actually say. So that is a very, very good resource if you want to understand more about GLP compliance.
Okay, what should be raw data archival timeline in a GLP lab?
So right now in the U.S., our GLP regulations state that as soon as a report is final, all of the data, all the raw data for that study needs to be archived. In actually the proposed revisions to the GLPs, I found a reference to 1 week being a guideline. But right now, where it stands, it says upon finalization. So you can put data into the archives before something's been finalized, especially for longer studies. If you have a protocol that's going to span several months, I would recommend putting the data into the archives piece by piece just to control it. It's important to note that once something goes into the archives, it's not like you lose access to it forever. It's just controlled so that if you need to access the data, you can certainly do so. You can sign it back out of the archives to use it for any kind of report writing or anything that you need. It's just a way to control it while the study is going on. So it adds a level of data integrity once you get information into the archives.
Okay, our next question. The GLPs seem generic in the way they are written. How does the lab actually implement them? How is it okay for company X to deal with a situation differently than company Y?
Okay, super. So yes, I agree. The GLPs are a bit generic in the way they're written, and I think that's a good thing. I think it's really helpful for the kind of the black and white line to be placed, but the way you get to that line is up to each facility. That's where test facility management can look at the requirements and determine the way that it makes sense for them to meet that requirement. So as long as the requirement is met, like in the example that I was just giving, as long as information is in the archives by the time the study is finalized, then it doesn't matter if company X puts information in weekly and company Y puts it in monthly, and company Z maybe waits until the report is signed off, and then that day it goes into the archives. All of those ways are still compliant. They meet the requirements. They just get there in different ways. And I think that's really important, especially where GLPs fall in a more research-oriented scientific way. We're not manufacturing things. There needs to be a little bit of wiggle room. And the GLPs, I think, are written in a way that it provides that wiggle room, but it also makes it clear who has the authority to make changes and decide when it's acceptable to modify what we're doing or not.
Okay, we have time for one more question. Is e-archiving required for GLP compliance?
So my classic quality assurance answer for that question would be it depends. If you are doing electronic data capture so that the first information you get is pulled off of, captured in software coming off of a machine, and then it's displayed to you in a spreadsheet type format, that would be electronic data capture. There are a number of things that go along with that data, like the equipment settings, the optical density, or if shaking was on, or the temperature, and oftentimes, as long as you're not using a generic login and password, you would also get the operator. All of that information, that's called the metadata, that's available as well and is tied to that electronic data. In that instance, then yes, you have to electronically archive that electronic data that you have. A paper printout of that data would not be sufficient to stand in for that data because there are pieces that are missing. If your data collection systems are a little more pen and paper and classic, so that you're seeing a reading on a screen and you're writing it on a lab notebook or pen and paper data capture, then there is no requirement that you then scan that information somehow and archive it electronically. That could be put in a paper archive that's secure. And also there's no requirement to do one or the other. You can have both. You can have both an electronic archive and a paper archive, and it may be very important and appropriate to have both. And there are a number of other questions that are still here. So I think, although we're out of time, what we can do is provide answers to those questions as well when we send out a copy of the slides, if that's okay.
So that concludes our question and answers portion. If we did not get to your question or you have additional questions, you can send an email to our presenter. Thank you for joining our webinar today. And we will be providing the slides and recording to this webinar, and we'll notify you when that is available.
And I just wanted to say one more thing. If you love compliance and you're going to be in the Washington, D.C., Arlington, Virginia area in January, I will be speaking at the second Good Laboratory Practice Compliance Summit. It's a 2-day meeting that'll be there. So I'd love to see you there. If you have any questions about that meeting, you can contact me.
And thank you for attending our webinar again. And again, you'll see on the screen if your question was not answered today or you do have additional questions, please feel free to email Mandy at the email address shown on the screen.
All right. Thank you, everyone.